In the 1880s, the great scientist called Charles Darwin and his son Francis discovered phototropism. They were very curious about how plants grow and move. In their experiment, they noticed something interesting: plants appeared to bend towards light. They wanted to know why this happened. They started their experiments with young plants, called seedlings. These seedlings were just starting to grow and had a special part called a coleoptile, which is a small protective sheath that covers the young leaves. Charles and Francis covered the tips of a few seedlings with little caps. They noticed that the plants stopped bending towards the light when the tips were covered. This led them to believe that the plant's tip was essential in sensing light. Then they chopped off the tops of some seedlings. Again, these plants also didn't bend towards the light, which demonstrated the importance of the tip in the plant's ability to sense light. They then used transparent caps to cover the tips. These plants still bent towards the light, showing that the tip needed to see the light to work properly. Through these trials, Charles and Francis Darwin proposed that the plant's tip detects light and transmits a signal to the other parts of the plant. This signal makes the cells on the darker side of the plant grow longer, forcing the plant to bend towards the light. This bending of the plant towards the light is known as phototropism. Phototropism allows plants to get more light, a clever mechanism for plants to move towards light, which they require to produce food through photosynthesis. After Charles and Francis Darwin established that the tip of the plant detects light and transmits a signal to cause the plant bend towards it, another scientist called Boysen Jensen wanted to find out more about the mechanism of communication. He conducted several excellent experiments in the early 1900s. He removed the tips of some seedlings, something as the Darwins did. But then he put a small piece of gelatin between the tip and the remainder of the plant. He observed that the plants can still bend towards the light source. This demonstrated that the signal could pass through the gelatin. Next, he used a tiny piece of mica instead of gelatin and observed that the plants did not bend towards the light this time. This indicated that the signal couldn't get through the mica. Boysen Jensen's research demonstrated that the signal from the plant's tip was a chemical that could pass through gelatin but not mica. This chemical signal was eventually discovered as a plant hormone called auxin, which causes plant cells to grow longer on one side, leading the plant to bend towards the light. This enables plants to get more light and create food via photosynthesis.